Hello, everyone. Welcome to the HKGI-APSHG Distinguished Scholar Series. My name is Brian Chung. I am the Chief Medical and Scientific Officer and the Interim CEO of the Hong Kong Genome Institute and the immediate past president of the Asia Pacific Society of Human Genetics. This lecture series is jointly hosted by the Hong Kong Genome Institute and the Asia Pacific Society of Human Genetics. Through this initiative, we aim to bring together world-renowned scientists, clinicians, and researchers to share their insights and experience in genomic medicine and genomic science. This is a special presidential series in which we have invited presidents of leading international human genetic society to promote global knowledge exchange and dialogue in genomic medicine. The series also serves to raise awareness of the upcoming International Congress of Human Genetics 2027. We can show that slide, which will be held from 1st to the 5th of March 2027 in Guadalajara, Mexico. Today, we're very honored to have Professor Martina Cornell, President of the European Society of Human Genetics, as our distinguished speaker. Professor Cornell is Professor of Community Genetics and Public Health Genomics at Amsterdam University Medical Center. She is a physician and epidemiologist. Since 2000, Professor Cornell has focused primarily on the responsible implementation of genetic testing and screening. She is a member of the Health Council of the Netherlands, where she serves two standing committees, population screening and the council group. She also chairs the Netherlands Program Committee for Neonatal Heel Prick Screening. Professor Cornell's research centers on community genetics, a field concerned with the responsible and realistic applications of genetic and genomic knowledge and technologies to health and diseases in human populations and community. It is a multi, inter, and trans-disciplinary field that seeks to maximize benefits, minimize potential harms, respect individual autonomy, and promote equity. Today, Professor Cornell will deliver the lecture entitled DNA in Public Health Screening Programs. Please join me in warmly welcoming Professor Martina Cornell. Martina, the floor is yours. Thank you.
Thank you very much for inviting me to present here. It's an honor to be here at your society, and I'm very happy to speak about this important topic of DNA in public health screening programs. I have no commercial conflicts of interest, no commercial relationships, but well, as Brian already told you, I am now vice president of the European Society of Human Genetics. Until a month ago, I was president, and I'm heavily involved in neonatal screening. For this upcoming hour, I will first tell you some things about the history of newborn screening, which is important. It is a public health intervention, often meaning that there is limited funding, and it's very important to have a high positive predictive value. So if a child is referred because of positive test result, it's really important to have a very high likelihood of really being affected. And then I'll speak a bit about the future of DNA in newborn screening and relevant European Society of Human Genetics statements in this field. And then I'll compare a bit the pros and cons as we see them today. And then I'll very briefly speak about other possibilities of DNA in public health screening programs. So neonatal screening is a public health intervention offered to all newborns for conditions for which effective therapy is available, because if you start that therapy early, it can prevent or ameliorate the disease. So those children with, for instance, hearing problems then can live healthier lives because they can be picked up early and treated early. And many of the conditions in the neonatal screening are genetic conditions. It all started with PKU, but for instance, also cystic fibrosis and metabolic diseases such as MCAD are often in the neonatal screening, but also hypothyroidism, which may be due to the missing of the hypothyroid gland, of the thyroid gland, so not always genetic. And what is important for today is that it's very diverse in different countries or even jurisdictions. In some countries, you have different health regions, and in one health regions, they may have a different neonatal screening program from another part of that country or another country. And that's not good for the children because then they may have different opportunities depending on where they were born. So it all started with PKU. Around 1950, the first treatment was available. Maybe the language is a bit different from what you would use now, but they spoke, for instance, about a crusade against mental retardation. The first diet turned to be effective in a child with mental retardation. That was a phenylalanine restricted diet. And then, of course, that works best if you start treatment early. So then Robert Guthrie developed a simple and inexpensive screening test that could be done before the child went home from the hospital, before it was discharged from the hospital. And that combination of having a treatment and a test made newborn screening possible. In many countries that started in the 1960s or 1970s. And on the left, you see a very real picture of two siblings who both have PKU, but the left one, the boy, didn't receive a diet, and the younger sister then received the diet very early onwards, and therefore is developing quite normal. And this treatment can avoid cognitive impairment. It's quite frequent, 1 in 18,000 newborns. It's a rare disease, but not that rare. And so this is what in many countries initiated then newborn screening programs. Which started at different moments in time. Here you see the situation in Europe, where some started in the 60s, others in the 70s, others even more recent. And in a few countries like Albania, we still do not have a newborn screening program. And this is the worldwide situation, where you see also the Asian and Pacific region. What is noticeable is that in some parts of the world where many children are born, such as Africa and India, still the newborn screening programs don't cover many diseases and don't cover all the children. In your part of the world, it's still very different. In some countries, you have good screening programs with many conditions and with a high coverage, and in some parts, it's not so very well developed. So there's still a lot to be done. And this map shows countries large if they have a large surface, if they have many kilometers, but not if they have many children. So when you think about the children born in these parts of the world, the situation is even less positive. Okay, so in many parts of the world, when they think about screening for a certain condition, they base their decision-making on the Wilson and Jungner principles, which were developed for the World Health Organization in 1968, but they were also updated in the 2000s. But if I summarize that very briefly, it is always about an important health problem. That's not always a frequent health problem, but it could also be PKU, which is already mentioned in that early document in 68 as important because there is such a big difference between being treated or not being treated. And then, of course, there has to be an effective treatment and a suitable test, which has to be acceptable for the population, but also good in terms of specificity and sensitivity. And there has to be an appropriate use of resources, which means that compared to other parts of healthcare, the healthcare expenditure has to be acceptable. That also makes the choices in poorer countries or richer countries somewhat different. Depending on the resources, it may not be affordable in some parts of the world, while it is in others. And if you summarize these screening principles in just one sentence, the benefits should outweigh the disadvantages. So that's very simple to remember. But if every country does this in its own way, there is a diversity between jurisdictions in the conclusions that they come up with, and that leads to not all infants in the world receiving the best care, and that's probably not what many of us want. Sickle cell disease and hypothyroidism, for instance, are quite frequent in Africa, but only less than 2% of the children are screened for these conditions in Africa, so that could be definitely better. And when you ask experts to evaluate which diseases need to be added to the newborn screening in their country, they may first of all think about the diseases that they are expert in. So we have to keep a bit wider view. It's not just metabolic diseases, it's also hearing problems, it's also, say, SCID, combined immunodeficiency, SMA, spinal muscular atrophy. So we really should try to think about the expertise of many different pediatricians. And it's very important to remember that neonatal screening is a program, and the program is more than a test. You have to inform the parents during pregnancy and just after the birth of their child that some blood is going to be taken and sent to a lab. You need an information system where the data on the child can be entered, and the results will be brought together with the data of the child, so that if there is a positive test result, you can trace the child, which then likely is at home. You need, of course, a pathway for telling the results to a physician who can then treat the doctor afterwards, often a specialized pediatrician. There has to be a reimbursement, not only of the test, but also of the treatment. So that is funding. If the treatment is not available, why would you screen? You need governance and policy making, especially when you think about adding more diseases to the neonatal screening and further improving the program. So there also it's important to think about who makes those decisions, who is involved. In the Netherlands at the moment, we screen for 27 conditions, most of them being genetic. And hypothyroidism is quite important, as I mentioned, but usually not genetic. And then every year we refer almost 500 infants, out of whom half have the condition we're looking for, but the other half is false positive. And that 50% that are referred but turn out not to have the condition, we like to have that figure a little bit lower. So we try to improve also that aspect of the program. And this entire program costs 146 euros per child, including the person going to the child when it is 3 or 4 days old, when children in the Netherlands often are already at home, and including also the letter saying that the result was good. So for the laboratory testing, it's only something like 50 euros, which is not very much. But it's important to remember that when you think about genome sequencing and newborn screening, which today in the Netherlands is much more expensive. And it's always good to remember that also some functional testing is done in the newborns, the hearing screening. For that, we refer 680 infants, out of whom 142 had bilateral hearing loss, so they really would not hear anything if that had not been recognized early and given hearing aids at a very young age. Now, when we talk about how good programs are, we often tend to mention the number of conditions in the newborn screening programs. But it's important to remember that for some of those, we will diagnose many infants, such as hypothyroidism, which is definitely in numbers much more important than many metabolic diseases, and sickle cell disease, which is quite frequent, and in more recent years, for instance, SMA. And in between, we added quite a lot of metabolic diseases, which are quite important, but each of them often has only a few cases that are affected. So they are important, and definitely when you do one lab technique such as tandem mass spectrometry, in 2007 we added 13 metabolic diseases at once because they could be identified in just one test. But it's important to remember also this number of children that will be recognized after adding another condition to the newborn screening. And then the test properties. When you think about a screening program, it is very important to have a very precise test. And the test properties might be a bit different from what we are used to in the diagnostic lab. When you think about screening 100,000 children, out of whom 10 would have a positive test result for PKU, and 9 would really have PKU, that means that the sensitivity is 100%, so we pick up all the PKU children, but the specificity is 99.998%, and this figure is much higher than for many tests in diagnostics, meaning that if your child is healthy, it will most likely have a negative test result. And the positive predictive value in this example is 90%, so that's quite good. But the specificity, just remember this, has to be much higher in a screening program than it is in diagnostics. So that was my short summary of the history. And then I'll turn to DNA and newborn screening and some relevant ESHG statements. It's important to remember for all the testing that the interest of the child has to be central when we evaluate what will be the next step in newborn screening. And this is from a paper in 2014 where we invited many policymakers in Europe to sit together and think about how to move on with newborn screening. This was not just Cornell writing about it, but it was really on behalf of a lot of experts from this field, representatives from government. And they said that always families may also be interested in having an early diagnosis to reduce the diagnostic odyssey, to avoid having two children with the same condition, for instance, Fragile X. But if it's not treatable, if the interest of the child would be harmed, then we definitely should not add conditions to the newborn screening. And something we discussed at that time also is that society may be interested because having an early diagnosis sometimes lead to savings. So there could be economic aspects, having more healthy people that can work, that can have a job, having parents that can go to work and not having to take care of the children. And also, if it is a rare disease and there's a lot of knowledge lacking, then we may want to generate knowledge via research. And that all is potentially interesting, but the interest of the child really has to be central in thinking about newborn screening. And then when an umbrella organization of parents in Europe spoke about newborn screening, they came to somewhat different conclusions. This is a document from 2021 from Eurodius. And of course, they also started from treatment and avoiding irreparable health damage being the most important goals of newborn screening. But they also stressed that for them, from the parents' perspective, avoiding a long diagnostic odyssey could also be very important, and limiting the number of unnecessary diagnostic procedures. Think about muscle biopsies in a child with Duchenne muscular dystrophy, or limiting the uncertainty and unfavorable psychological effects. Telling your son that he has to try to walk, for instance, and if after a while he turns out to have Duchenne, then perhaps you would want to give him other kinds of advice and trying to give him physiotherapy to keep his joints mobile instead of saying you have to walk. So the psychological aspects for them counted heavier than for the medical experts. And they also mentioned that for them, the benefits of the family and society could count into the evaluation of pros and cons. What is anyway happening is that more and more conditions can be added to newborn screening programs because more treatments are becoming available. First, it was about diet in PKU and medication, thyroxine and hypothyroidism, lifestyle advice for MCAT, having frequent feedings, but more and more also stem cell transplant and gene therapy, often possibilities for those rare genetic conditions. And another development is that more tests are becoming available. Tandem mass spectrometry made it possible to screen for many metabolic diseases all at once, one test for a whole set of conditions. And PCR and DNA testing also make it possible, of course, to test for many genetic conditions with just one test. And then some countries also consider broadening the goal, also adding actionable conditions as the Eurodius document is defining, raising the question whether research can be a goal of neonatal screening. Anyway, for genomic newborn screening, for the moment, the answer has to be yes, because there we see that all the pilots are research pilots, and they will make us learn a lot about potential aspects of genomic newborn screening. In the traditional newborn screening programs, already DNA is often a part of the program as a first tier, as a first step for SCID and SMA. In SCID, the combined immunodeficiency, the first test is a TRAC test, the T-cell receptor excision circles, which is DNA. And then for SMA, countries look at the copy number of exon 7. If exon 7 is lacking, it means that a child has no active SMN1 gene. And in the clinic, then also the copy number of SMN2 will be assessed, because if it happens to have many copies of SMN2, then still the symptoms may be quite limited. But if it has 1 or 2 copies of SMN2, then the clinic will be quite serious. And then sometimes DNA testing is combined with other tests, like for cystic fibrosis, where first of all IRT is often assessed, and later tests look at the gene itself, either a limited number of mutations or sequencing the entire gene. And one of our recent additions in the Netherlands, adrenoleukodystrophy, we just want to report in boys, and therefore a DNA test for the number of X chromosomes is added, and gene mutation analysis, because that way we are sure that we just report pathogenic variants in boys. Now, when we spoke about 10 years ago about the future of newborn screening, of genomic newborn screening, a cartoonist has been drawing this cartoon, a stork bringing the new baby, and the vulture bringing the genome card, which definitely shows that not everybody felt that this was such a good idea as yet. But, there could also be a lot of cons in bringing a genome card for a newborn. But anyway, after that, a lot, of course, has happened. So the future could certainly be that DNA testing will make it possible to test for more conditions. And if in your country you think about adding more conditions, you may want to look at tests used in other countries and first of all add those that have been tested in other countries. You also may think about testing treatable monogenic conditions for which we don't have a biomarker now. So we can only add that if we do have a DNA test. An example is cystinosis. You may also think about congenital infections because viruses also have DNA or RNA. If congenital CMV is identified early, the consequences like vision and hearing problems can be limited. And conditions for which the scientific evidence is still limited, but clinicians are convinced of the pros. And there we need to, of course, then have research also as a goal of newborn screening, because the scientific evidence then still would have to be generated once we have started the newborn screening. And we may think of conditions that are actionable in different ways, although you may consider there to have informed consent at a different moment. When you think about Duchenne and Fragile X, as the parents' organizations mentioned, for them quite important, but for many traditional newborn screening programs, not a part of their programs yet. So in theory, it would be possible to move from something close to 30 to something close to 300 conditions if you use next-gen sequencing. And then thinking about the pros and cons, some mutations might not be leading to symptoms in childhood. Although we think we know all those genetic conditions quite well, we tend to know them from the clinical cases with symptoms. But identifying children with two mutations might also lead to overdiagnosis and false positives. So therefore, many countries consider that they still want to depart from evaluating pros and cons per condition and have pilots to generate evidence. So to first study what happens if they start using DNA testing. And then again, from the ethical perspective, as long as the interest of the infant is not harmed by an early diagnosis, advantages for the parents, for the families, for society could be evaluated in that first evaluation of pros and cons. But the child can be at the center, even if also parents or society profit to a certain extent as well. Now, one of the programs that has been developed early and I want to talk a bit longer about is the Newborn Genomes program in England, the UK Generation Study. It builds on the ambition that in the UK they want every child to be able to receive their whole genome with the U-prick test in due time. And for this purpose, a list of more than 200 conditions has been selected. Again, every condition has been evaluated. And for each of those conditions, more than 500 genes have been selected linked to those conditions, and more than 100,000 newborns will be sequenced in this UK Generation Study, which is a pilot. And the principles for inclusion of those conditions look quite a lot like the ones that we have discussed just now. The Wilson and Chung criteria are, well, what has been elaborated from that. They are still looking at variants that cause the condition, strong evidence. They are looking at a high proportion of individuals who would expect to have debilitating symptoms, so a high positive predictive value, if you like. There needs to be an early or pre-symptomatic intervention which can improve the outcomes in children. And the treatments, the interventions have to be equitably accessible, meaning that if it's available in part of England, but not in all England, then it has not been added to this list. So if the treatment, for instance, would be too expensive or the medical expertise is lacking in a part of the country, that is a reason not to report the variants in the generation study. Thinking again about how you would then develop from the idea of expanding to sequencing the list of target conditions. Again, we need those three steps of first the target conditions, the diseases, not just screen because you can, but really think about each condition separately. They need to have symptoms in childhood. They need to have treatments or actions available. They need to have a high penetrance at a young age, and therefore we need to avoid patients in waiting. Think of Pompe. I know in some parts of Asia you are screening for Pompe, but in several European countries we have a lot of mild Pompe cases. And it's not so easy to make a distinction between the serious cases in childhood and the patients in waiting that would only develop symptoms in adulthood. So yeah, the list of target conditions is the first thing. And then within each of those conditions, the list of genes for analysis. Again, making sure that the genes you analyze will lead to treatable cases. And then the list of variants within those genes is important. You might think of just class 5 or 4 genes and not variants of unknown significance. Although, again, from an equity perspective, the variants in the Asian populations might be less known than in the European population. So, if you would have a combination of DNA testing and an orthogonal test, say an enzyme test or a metabolic test or another biomarker, then perhaps you would want to report more than this. But usually, if you just think about DNA testing, stick to the pathogenic variants. When you speak to the people in your genome lab and you say, I want to start a pilot of genomic sequencing in human screening, and I want to first do a validation, then they may say, oh, but we validated our gene technology very well already. The analytical validity is very good in our lab, but still the validation in the context of population screening needs to be a little bit different because the questions are different. And here at the bottom, you see false positives or patients in waiting. And that's related to the question, does a child with 2 pathogenic gene variants indeed develop clinical symptoms in due time? And when we started expanding our traditional newborn screening programs with, for instance, biotinidase deficiency, we thought we knew the condition quite well from the clinic. And after the start of the screening, 10 times more children with biotinidase deficiency were recognized. And with galactosemia, the same happened. Because definitely we did not know the condition completely, especially the mild cases or the non-symptomatic cases we did not know. So an important question is, could 2 pathogenic variants lead to a still sufficient enzyme activity? And are second-tier biomarkers available for confirmation? Because 2 mutations and a still sufficient enzyme activity is perhaps something you would not want to report. And already for some conditions, we know that there are milder and more severe phenotypes. So you might want to develop a test that can only report severe phenotypes, or a combination of tests, 1 DNA test and 1 biomarker test. So this is about false positives. And then if you think about whole genome sequencing in newborn screening, you also need to think about false negatives. Perhaps you would miss conditions that cannot be detected with whole genome sequencing. So if you would replace the complete program with only DNA testing, you would definitely miss part of the congenital hypothyroidism cases, for whom it's very important to have early thyroid treatment. Some countries screen for vitamin B12 deficiency, and that's often not a genetic condition, but also just caused by the diet of the parents. So you can miss diseases because they are just not in the DNA. And then you can also miss cases because they don't have 2 known pathogenic variants, but the same condition would still be at stake because perhaps there's other unknown variants, or perhaps there is another mechanism. Think of an epigenetic mechanism. So the combination of a biomarker and DNA testing might avoid this kind of missed cases. So the analysis of the analytic validity is not sufficient, again, at the level of the false negatives. Thinking about DNA screening in 2026, we have many traditional newborn screening programs that already incorporated DNA testing, which helps to have a very high specificity. And we now have many pilot projects using whole genome sequencing in genomic newborn screening, but these are pilot projects. So now I turn to the ESHG statements that could be relevant in this domain. And 3 of them are quite important, but are from the period 2013 to 2016. So you may think they are old, but I will argue that they still are relevant. And I'll also refer to Wilson and Chung, which is even older. But thinking about why we wrote them in that way and what is the relevance today is quite useful. So the first one I'll speak about is the whole genome sequencing in healthcare document of the European Society. That first spoke about if we start doing whole genome sequencing or whole exome sequencing at that time, We really have to develop best practices, think about what we can learn from our early experiences and share those experiences with other countries and establish testing guidelines. So that's what we have been doing, and we have been learning a lot from that. But we may want to do that again if we apply those techniques in newborn screening. And when we summarize this document, we often said it's about target, target, target. Target the conditions, target the genes related to those conditions, target the variants related to those genes, and that we know quite well. And in that same document in 2013, we already said that in screening, the use of genomic information often calls for a different standard of evidence thresholds because, as I showed you, the evaluation at the population level might require different things like a very high specificity. And the challenge will be to avoid a broader scope in the test that is not based on the evaluation of clinical utility and other screening criteria. That's what we wrote 10 years ago. And already Wilson and Jungner also have been warning in their introduction to their document. They already say that with so much curative work to do for medical doctors and little time, little resources, we may want to, first of all, treat people that are sick, and we may not want to spend our time or money looking for diseases in its incipient stages. So that's already a warning. If we have limited resources available, think about whether this really is saving time and leading to better healthcare or not. And then Anderman in 2008 had a look at the Wilson and Jungner principles again in the age of genomics, and they repeated all the old issues like serious health problems, suitable test and treatability, but also added that there should be scientific evidence of the screening program effectiveness. So it's not just the screening test, but really the program effectiveness. And what he also mentioned is that it's important to ensure informed choice, confidentiality, and respect for autonomy. So they stressed more some of the ethical principles. I'm turning back to the European Union developments around 2014. As I mentioned already, we had some important stakeholders sitting together then. So the geneticists, but also the EU member states, people who were involved in the screening programs then. And they also wrote that the interest of the child has to be central. They wanted to avoid unintended findings. They stressed that we should keep analyzing per condition, not per technique. And they stressed that it's important to have information. The provision of information needs to be organized at the program management level. So public health authorities are responsible to do that so that all the relevant users can really see that information. Don't just think about a test, think about what the public will read about it and parents that potentially want to have their child participate, the information that they need, and the physicians that will see those patients after referral, also those relevant stakeholders, the pediatricians, need good information. And then we wrote a document in 2015 specifically on whole genome sequencing in newborn screening. Many things are repeated that we discussed earlier. Targeted analysis for conditions for which the treatment has to start in the newborn period. The evidence base has to be very good. The infrastructure and the human resources needs to be there. And that's not just the testing, but also the monitoring, the education of the physicians involved. All costs involved have to be part of the evaluation of the public health program. And what was important then is that they stressed the importance of having an open dialogue about all the expected medical and social benefits with all stakeholders. It could be the patients and patient organizations and the general public. And then we didn't speak about newborn screening and genomic newborn screening for 10 years, perhaps because this was quite clear what we had said 10 years ago, and people working on it could read those older documents. But this year we spoke, last year actually, we spoke about newborn screening in Greece, where we called for openness and transparency, because after a small pilot, they were planning to have all newborns screened with genomic newborn screening in a few years' time, with their parents not asking for consent and sending all the materials to a company in America meaning also that the data would not be in Greece anymore, but would be in a company in the United States. And some people came to the European Society saying, is this really the way we have to move on? And we called for openness and transparency. And after this call, indeed, the government stopped the process a little bit and, first of all, tried to speak with all the stakeholders involved. So that's probably going to be much better in due time. Again, coming to next-gen sequencing, which as a pro definitely will make it possible to screen for more conditions. But for each condition, we need to evaluate pros and cons. And we need to think of the overdiagnosis and false positive and all the potential harms, balance the pros and cons. When you think about arguments against screening in general, already Moya Gray, who was the director of the English screening programs, warned, not in the context of DNA, but very general, that all screening programs do harm. Some do good as well, and often some do more good than harm at reasonable cost. So this was not a document about genomic newborn screening, but just about any screening. Screening for prostate cancer could be true as well. It might harm, and then if you look for the resources, the reasonable cost, then many programs that would screen are quite expensive as compared to regular healthcare. So this is a statement of more than 25 years ago, and for next-gen sequencing, we have to remember this. And then we have to think about the possible risks, for instance, false positive, false negatives, medicalization, saying to a person, you carry 2 variants in your genes for a certain condition, which means you are a patient, or perhaps you're going to be a patient, and the rise of the pre-patient, meaning that people are told that they are a Pompe patient, but perhaps will not develop symptoms for the first few decades, and that's perhaps not what we want. And something different is that it might undermine the autonomy. If people say, we are all participating in this genomic newborn screening, parents may feel that there is a kind of a social pressure for everybody to participate, which is perhaps not what we want at this point in time. And then there might be disproportionate worries, again, linked to the finding of some variants that we cannot really tell whether they will lead to a certain disease. So what we need is the population prevalence of variants. For Asia, this could be even more important than for European or American countries. The population prevalence not only in sick, infected persons, because that's what we often know relatively well, but also in conditions in newborn screening or potentially being added to newborn screening, because then we are speaking about the entire population. And we need that in different parts of the world. It might be quite different in one country in Asia from another country. And then from those two, the variance in affected persons and the population prevalence, we might estimate the penetrance of combinations of variants, the likelihood of really developing symptoms. And then for newborn screening, I said in summary that it is definitely possible to screen for more conditions, but we need to proceed with caution because there also are some potential disadvantages, such as the overdiagnosis, the positives, the false positives, false negatives, and undermining autonomy. Now, so far, I have only spoken about newborn screening. Just very briefly, it's also possible to think about DNA in other public health screening activities. If you think about BRCA or Lynch or familial hypercholesterolemia, if you identify one patient because they have already symptoms, you would want to identify the family members who are still healthy, because for those first-degree family members, for instance, prevention is often possible. And nowadays, not all relatives are informed. So you could, in a very systematic way, also organize a public health screening program for the first-degree relatives of the identified sick people with those conditions. But now often that is not organized in a systematic public health program. And the same might be true for pharmacogenetics, where nowadays we sometimes test patients for a certain relevant variant, but not always. And we also don't always put that in the electronic health records. Now, if, for instance, a person is needing certain chemotherapy, fluoropyrimidines, often the PYD gene is tested because some people may have serious side effects, but that DPYD status needs to be also then in their electronic health record. And if you do one pharmacogenetic test, often it's very cheap to do a wider pharmacogenetic test. So you may want to register those pharmacogenomic variants in their health records. So the next time they need a certain drug, the physician can be warned that these people need a different dose or a different drug. And that also could be a public health approach if you really put that in the whole health system. As European Society, we had quite some activities on DNA in public health screening programs last year. We had webinars in January and February, and they are still available on the YouTube channel. So if you want to listen to them, even if you did not attend at that moment, they are still there and you can still watch them. And then we have a special issue of the European Journal of Human Genetics on DNA in public health screening programs. You can't submit anymore, but there's already more than 25 papers that can be found online, which also cover the broad scope of the things I talked about today. Many genomic newborn screening, but also a few pharmacogenetics, for instance. And then I hope that next year at our European Human Genetics Conference, we will again speak a lot about genomic newborn screening and DNA and screening programs in general. And I hope to see you all there, but of course also in Guadalajara. And if you have any questions, then I'm now happy to answer them.
Thank you very much, Martina, for the great talk. A very good revision about the history of newborn screening and how it has been and is still the most impactful genetic program around the world in terms of the impact on human health. And thank you for also zeroing in to talk about how the testing of DNA from genetics to genomics has influenced the practice. I think it's just great to see that the criteria proposed by Wilson and Younger so many years ago is still holding true. So it's really reflecting the intelligence and the smart ideas that has been put in this criteria, so that it's almost like the bulletproof even for decades. Another amazing thing that I have seen is the great work done by the European Society of Human Genetics in all those position statements that helps to clarify and modernize the concepts and how we can put those concepts into applications. And of course, the ongoing support in professional and public engagement and education on important topics such as DNA in community health level, including newborn screening. Thank you very much, Martina. So I will pass on the questions and answer sessions to Wei to host. But Martina, can I start by asking you for your comments? I attended, as I have told you, the recent European Society of Human Genetics conference, and there are multiple good sessions on genome newborn screening. And I see now the level of evidence, although it is still at a very beginning level, has already switched the concepts of a lot of people that is involved in the field from shall we do genome newborn screening to how do we do genome newborn screening. What is your personal view on this? May I ask this first question? Thank you.
Yes, I also saw that 10 years ago we asked the question, should we do newborn screening? But more and more the evidence is quite strong that genomic newborn screening definitely has potential. So the question now is more indeed, as you say, how? One difficulty I see still is that the world of genetics and the world of public health are very separate. So we need more contact between those two worlds to really talk about what we can learn from each other. Because, for instance, the penetrance, people in the genomic labs often think that they know already the penetrance, and when they start doing a population screening program, it turns out to be somewhat different. And the pilots, of course, are very good for that kind of experiences, for really being forced to collaborate with all the relevant stakeholders. And if you like, learning the childhood diseases, or well, learning the first lessons from, if you start to really make this into a public health program. And in the beginning, that is quite expensive. And that takes a lot of efforts to learn the lessons. But many of the lessons can be used all over. So they are not just for one country, but they are for many countries around the world. So we need to also listen quite carefully to the lessons that they are learning in those pilots. And after that, I really feel that we probably could scale up many of the newborn screening programs from, say, 30 to 300 conditions. Although there are lots of questions like, can it be done in every country? Are the treatments available? I mentioned SMA, but in the Netherlands, we added SMA once the treatment was available, once the gene therapy became available, and not when it was an untreatable disease, of course. So many of those questions keep relevant, and we need to proceed with caution, but I still think we will proceed somehow.
Thank you so much. I'll pass the time to Dr. Ma to host the question and answer sessions.
Thank you, Professor Connor, for your very inspiring talk. We have a questions in the chat box. I will. The first one is like regarding the time that we needed to like analyze the variants detected in the genetic newborn screening, whether they are pathogenic or lightly pathogenic. Do you think, or do you have any recommendations about whether there are like tools being developed to assist this process that will help to reduce the time that's needed to complete the report, especially considering it need to be high throughput if it is being implemented in a population screening scale. So do you have any good solutions to make or to even to further shorten these turnaround times for the newborn screening and genetic reports?
Yeah, so I don't have those solutions myself, but I know that, for instance, in England, they're also thinking about this question that now it is a pilot with good funding, but if you want to make this into a screening program with little resources, you really need a pipeline that is just reporting the combinations of pathogenic variants, so just children with two pathogenic variants. I don't know whether it's ready yet for global use. So for now, it's still, from all the pilots where I've been hearing about, it's still a lot of time and resources needed. But yeah, say between now and the next few years, I think it will really be pipelines where just the ones that are very specific will be reported. Maybe it's also good to mention here that I know about project in the Philippines where they try to build a different kind of an array where you have on the array just the variants that we know are pathogenic in that specific population. And in that way, it would be a cheap and perhaps effective test, although not with 100% sensitivity, but for a screening program, that's not always needed. And that's also still under evaluation. So I don't know how that will develop over the time, but not just doing the whole genome sequencing at high throughput at expensive machines, but also thinking about arrays is another way to think about this.
The second question is asking about your opinion on, beyond the newborn genomic screening. This audience is asking about the potential of prenatal screening, such as screening samples like amniotic fluid, whole genome sequencing, or a targeted sequencing panel, being used in prenatal screening programs.
Yeah, so one difficulty of the prenatal setting is that then if there is not a phenotype, then it may be very hard to interpret the variants. So about the NIPT, the non-invasive prenatal tests, often for chromosome anomalies, and there the predictive value of those variants of those phenotypes is quite high. But if you would add many conditions to such a screening, then you might also come into situations where you predict something where the positive predictive value is much lower. So doing that for children with ultrasound anomalies is still somewhat different because there you know that there is a certain phenotype and you use similar techniques to find the cause of the ultrasound anomalies perhaps. But doing that really as a screening is still quite challenging at this point in time.
Thank you. And the next question is, how can we distinguish variants that cause severe childhood diseases from those that are associated with a relatively mild and late onset or even non-penetrant phenotypes? And how does this information helps when designing a genomic newborn screening panel?
Yeah, from my point of view, there a very important strategy would be to look for two tiers or more tiers. One could be the DNA test. One could be, for instance, a biomarker. I mentioned the metabolic diseases. If there you would have also a test for enzyme activity, And the combination of first a DNA test and second a test of enzyme activity or metabolites or whatever might make the specificity quite good. But I mentioned the example of cystinosis where we have no biomarker, and then of course it's more difficult. But this combination, from my point of view, makes things very feasible.
And I think there is a follow-up question regarding this one is because nowadays we have many large-scale genome projects all over the world. If like when considering variants with reduced penetrance or like variants that's related to a relatively mild phenotype, do you think that like these genome projects, the data from them can contribute to the newborn screening projects, even in the design stage, not contributing after receiving those results from the pilot?
Yes, very good suggestion. Because I mentioned two types of data that we need, the variants from the healthy people and the variants from the affected people. And the variants from the healthy people might already be in those big data sets. England has done a few of the studies where they try to look at the penetrance of some conditions in their genomic newborn screening program. And a few of those papers are already in the special issue that I referred to. So there they, first of all, look at Genome of England genotypes and what they would predict in terms of the prevalence of certain diseases. And then they compare that to what we know from other sources. And they see that, the predicted penetrance then is much lower than we had anticipated some years ago. And that type of studies is also, of course, possible in different parts of the world. If you have big Asian databases with variants that are relevant in your parts of the world, then you could combine that with the knowledge you have on affected persons. And if there is a big discrepancy between variants, people being, having two variants in their genome for a certain condition but not being affected, and that also tells you already, even without doing the newborn screening, that the genomic newborn screening might lead to many false positives. And it might also give you clues where to look for a second tier. If some of the conditions have a very high penetrance, then you might not need a second tier. If some of the conditions have a high number of false positives, then that's the ones where you would not make a screening program without a second tier.
And we have another question related to the evaluation. So this audience is asking, beyond the traditional clinical outcomes, what frameworks should we adopt to evaluate the broader impact of the newborn newborn genomic screening programs, including psychosocial, ethical, economic, and health system effects, and over what timeline should these outcomes be meaningfully assessed? Will it be like short term after the participants reach their childhood or even into their adulthood?
Yeah, that's a very important question because the evaluation should not only integrate the medical aspects, but for instance, the false positives, the psychological aspects are quite relevant. And those could be very favorable for the children that have been identified at a young age. And this could be less favorable for children that are patients in waiting, for instance. And by default, this needs a longer time horizon. So if you think about the psychosocial aspects and also the economical effect, that really needs years as follow-up. Again, there you may want to look at the pilots that are now ongoing and hopefully will have a longer-term follow-up. But often you have to really think about 5 years, 10 years, or that kind of time frames to evaluate these things properly. If you start developing a program for your country, you may also want to speak with the stakeholders in your country, what they consider very important aspects to integrate in the evaluation, because again, that might differ from country to country. If in a country where the resources are somewhat limited or the family structure is much more important than the individual autonomy perspective that we often have in Europe, perhaps you have sometimes a different stance on what would count for you for another country than, say, England. It's also something to discuss early within the country you are thinking about pilots, because some aspects, as I said, might be different.
The next question is asking about the approach that we expand the number of conditions being included in the test. So as the genomic newborn screening expands, how should we prioritize conditions, treatable, actionable disorders, or conditions that may benefit from earlier diagnosis and management? So this audience is like asking, shall we follow the a broader panel like the UK's, or focus on conditions that are more prevalent and clinically relevant locally?
Yeah, I think definitely you have to look at what is more prevalent locally and more important to treat locally. Many of the pilots worldwide, for instance, report G6PD deficiency, and that could be a very relevant question for you to discuss in your part of the world as well. It's definitely more prevalent. You might say it's often not so serious because, many of the people with G6PD might not have very serious complications, but some of them might. And if you report that very early and you have good healthcare for these infants, a pediatrician who can explain what is the risk, what is the drugs to avoid, what is the food products to avoid, then perhaps a very simple intervention could be worthwhile. But now many of the pilots that are reporting G6PD just started doing it and didn't really think a lot about what could be the advantages or disadvantages. So just copying the English list is not a good way, I think. Copying the English structure to first go through a list of all the conditions, but they are prioritized the treatable conditions that are quite frequent in your part of the world might be a better approach. So the systematic way of going through all the potential conditions and the variants that are relevant for those conditions and the variants that you would report in your part of the world, that's definitely a very good structure. But then the list might be a bit different from country to country because some things might be more prevalent and more relevant in your part of the world than elsewhere.
Thank you. I think we can have a last question. So this audience is asking, will the advance of newborn screening and rapid genome sequencing, we are now able to diagnose like many rare disease patients, even in the first days of their lives when they remain pre-symptomatic. How close are we to a system where these diagnoses can trigger the development and deployment of the, maybe a targeted gene therapy before the symptoms onset? And what do you see as the primary obstacles to close this gap between the speed of our diagnosis and the personalized and targeted therapeutic intervention?
Again, a very good question, so thank you. The turnaround time is a very major obstacle at the moment because many of the turnaround times in the pilots are still rather weeks than days. So having a very fast diagnosis in a few days is often not possible for the whole genome sequencing applications nowadays. When you think about a sick child in the clinic, there is sometimes that rapid whole genome sequencing, but that's definitely not in the newborn screening programs today. So many of the older traditional newborn screening programs are much faster in recognizing conditions very fast, say in the next few days. So that's important to remember. And definitely the gene therapies are, potentially more important over the next decade. And there, from my point of view, the important obstacle is bringing those to the market. So we need, either companies to produce those medications and make them available with market authorization and also reimbursement. But for now, some of those diseases might be too rare. So perhaps we need academia to develop some of those gene therapies without a very high price. And perhaps we need our ways to bring those to the patients, which are even more personalized, perhaps in a platform way where we can develop for each patient their individual therapy, not being hopefully so very expensive.
Thank you very much, Martina, for giving us once again a great lecture. And I think the richness of the Q&A sessions and the discussion shows you that the topic that you have introduced to us today is really in a lot of people's thoughts. And many people among the audience have given a very serious thought about genome newborn screening. So obviously, this is a very hot topic. Thank you very much once again for the excellent lecture. And everyone, now you know that next year, ESHG meeting is going to be in the Netherlands. It's a great country to visit. Let me also do some promotion. So this lecture series will continue. Now we have heard from the president of the European Society of Human Genetics. I just want to let you know that in September, we'll hear from Ambrose Wonkum, who is the president of the African Society of Human Genetics. So this is the next lecture. Please pay attention or scan the QR code. And once again, I want to lastly show you the flyers from the International Congress of Human Genetics. So this is a joint conference between all the continental societies of human genetics. And those are the dates, the deadlines that you may want to pay attention to. And of course, the QR code will help you. With this, thank you very much. See you next time. Thank you so much. Martina, thank you so much again for your great talk.
It was a pleasure. Thanks for inviting.